Right. So we blazed through talking about photosynthesis and carbon capture. So let's pick up here. Let's talk about photosynthesis as it relates to nitrogen, and then hop into a couple other element cycles. Photosynthesis, of course, is broken into two big cycles. So photosynthesis is not just a process of just capturing CO2. First, it's capturing light energy um, from the sun, using that energy to generate some intermediate products that then move us into um, the dark reactions of photosynthesis where CO2 is actually fixed. So this involves enzymes, one enzyme in particular that is going to fix CO2 out of the atmosphere or out of the stomatal cavity where it is, and then turn it into those first three carbon compounds in the case of C3 photosynthesis, maybe a four carbon compound in the case of C4 photosynthesis. And here's where the interaction between cycles really becomes important to focus on, because rubisco, which fixes that CO2, is very nitrogen hungry. It loves nitrogen. And so we brought this up before, we'll just reiterate it. This enzyme really needs nitrogen. This is the single largest enzyme draw for nitrogen, and it's a reason why nitrogen is closely tied to the carbon cycle. Of course, we need phosphorus too, and we were talking about those intermediate compounds for transferring energy from sunlight and then moving it into the dark reactions. These are phosphorus compounds that are doing this. Phosphorus is important for growth, cell division, DNA, RNA, the backbone of DNA and RNA, energy transfer, active transport nutrients. We've talked about that some already. For all these cycles, we really need to think about fluxes and pools of elements. And we've talked about that again, so this is just a general review. And that's important. Let's jump ahead a little bit and let's talk about nitrogen. We know these are data, again, from Eon soils, from soils around Evergreen, where you can see a close coupling of that carbon and nitrogen relationship. And this is a perfectly accurate graph of the entire nitrogen cycle. It's very complicated. And it's amazing what you can find when you just do searches for nitrogen cycles. And so I found this one, which I like. It's a little cartoony, and it's missing some key pieces. Okay, maybe a little too simplistic. I'm also not so sure why the person has to be a skeleton. And then it shows amino acids. So anyway, maybe we better get a little more complex. Let's go here. Okay, here's a version of the nitrogen cycle that's maybe a little more familiar to you from your reading. In this case, you can see inputs from N2 gas, which is in the atmosphere, come in the form of, it can either be fixed by lightning, it can be fixed through the Haber-Bosch process, which is an industrial process. It can actually be, or it can be fixed by organisms. And then we could think of it as coming into storage form, coming into form as ammonia or ammonium. But there's some of this that, there's a little complexity here that might be over the top. My favorite depiction of the nitrogen cycle these days is this one. So wonderful, wonderful image. And actually remarkably accurate. So we start with nitrogen fixing nitrogen in the air that can be fixed by bacteria, lightning, or the Haber-Bosch process, which creates fertilizers with a lot of energy and pressure. Lightning will generate some fixed ammonia. It will break up that triple nitrogen, triple bonded nitrogen molecule in the air, N2, and break it apart, turn it into ammonia or nitrate. Once that happens and it's fixed into the soil, there are those two main mineral forms, ammonia and nitrate, that represent nitrogen. We'll talk more about those in the future, but there's a whole cycle associated with where those go, which organisms get to choose ammonia or nitrate first, which ones have priority kind of consumption of those, and then there's a process by which nitrogen can be internally cycled within an ecosystem. And then, of course, lost. And when it's lost, it might contribute to things like eutrophication of water bodies, or it can be denitrified and sent back up into the atmosphere. So here's another version of this, another kind of cartoony version, but this one actually has some values. So in this version of the nitrogen cycle, you can see actual values for both marine and terrestrial nitrogen fixation. You can see the relative importance of biological nitrogen fixation versus fixation through other means, or contributions to atmospheric nitrogen through human activities and industrial activities. So lots interesting going on there. Point out a couple things here, and the first, probably the most important thing for terrestrial ecologists to realize with regard to the nitrogen cycle, beyond the dynamics of the nitrogen cycle, which we'll talk about later, is that if you look at natural biological fixation, fixation by lightning can occur, but you can see the values here are relatively small compared to biological fixation. And then look at the amount of nitrogen that's added to the atmosphere through human activities or denitrification or pyrodenitrification through fires, or denitrification that can happen from human activities. Humans, we know now, have approximately doubled the amount of nitrogen coming into ecosystems, and similarly, we've contributed tremendously to nitrogen in the atmosphere. So we've really increased the nitrogen cycle, both the amount that comes in and the amount that goes out to the atmosphere. Phosphorus, on the other hand, is a really different kind of cycle. And so if we think of the nitrogen cycle in all its complexity as having a really limited amount of input, that is, the amount that comes in any given year might be limited, and it cycles internally and then goes back up into the atmosphere. There's always a pool from which to draw from in the atmosphere for any ecosystem. And so this is this great irony of nitrogen. It's all around us, yet it becomes limiting for ecosystems because there are few organisms that know how to break the triple bond nitrogen molecule in the atmosphere and turn it into something usable. Those first usable forms like ammonium and nitrate. On the other hand, phosphorus is totally different because there's not an inexhaustible pool of it, and you're not just waiting for the right organism to come along. Instead, there's an exhaustible pool of it. Phosphorus generally comes from mineral sources. It's coming out of rocks. It's coming out of formerly igneous material, and then it needs to be eroded and decomposed from that rock in order to become available. In this graph, which is a classic graph from 1976, what we call a citation classic paper that really examined the phosphorus cycle in an ecosystem and what you can expect through time. Soil age is on the x-axis. So as the soil gets older, you see that it starts out with a high amount of mineral phosphorus. But that phosphorus is not immediately available to organisms because it's locked in rock. It needs to get cleaved out of that rock, and then it becomes organic P, or it becomes phosphorus that is available for organisms. Some of it gets occluded, meaning that it is kind of sequestered away and it's not available for organisms, either because it's maybe because it's chemically bound in other substances in the soil. So just a fraction of what gets broken out of that rock becomes available to organisms. That peaks and then through time declines. So that in a very old system, we expect phosphorus to have disappeared a little bit. It's eroded away, it's weathered away, and there's less phosphorus available for organisms. This has been found to be true in systems like the Hawaiian volcanoes, where you can go and you can look at islands that are different ages. They've risen above the sea at different ages, and the older islands are more phosphorus limited than the young islands. Similarly, if we go into forests of the Amazon basin, we tend to find phosphorus limited soils, heavily weathered soils where phosphorus has been leached out and the rocks have been eroded enough that there's no longer a lot of phosphorus around. Whereas when we go into young systems like the forests around the Andes or the Cascades, where there's been recent volcanic activity, we have less phosphorus limitation. And we generally think of those as nitrogen limited. Okay. For both of those cycles and for carbon, we're going to spend more time going through them, but this brief introduction at least brings us up to thinking about cycles, about different sources and different processes that make elements available to organisms through time, and there's a change through time. So just the same way we think about biomass changing through time, availability of nitrogen might change through time as it follows carbon, and availability of phosphorus might change through time as phosphorus sources get eroded away. If we think of a system in terms of its successional stage, there are some generalities that we might be able to make, and these have been made again and again in the literature. Odom, in the classic 1969 paper about a system's perspective, really popularized the notion that as forest systems get older, they reach maximum biomass. And along with that, we might think of maximum ecosystem processes taking place with regard to things like water storage capacity, or the amount to which a system changes infiltration. The erosion potential of soils in an older system is greatly reduced. So the beneficial hydrologic functions increase in an older forest. An older forest also stores our maximum amount of carbon. So we put in the most amount of carbon in the bank. That older forest also, even though the soils may be weathering, they're weathering on a longer time scale than the time scale we're thinking about in forest succession. So in a phosphorus, availability sequence, we might be thinking about millions of years, at least thousands of years. And here in forest succession, we're thinking about hundreds of years. And so older forest has accumulated in the biomass the maximum amount of carbon, nitrogen, and probably phosphorus. And that'll all be cycling internally. A young system, by comparison, is behaving more like an open system. So there might be sources of phosphorus, but there's not much in the biomass. There are sources of nitrogen in the atmosphere, and nitrogen might be being fixed very rapidly, but there's not very much in the biomass. And carbon doesn't have much in the biomass. So in terms of water function, carbon function, nitrogen, and phosphorus, all of these are really maximized in an older forest. But this is also dependent on disturbance frequency, and it's dependent on recovery rate following disturbance. So a highly disturbed system can behave a little bit differently. Let's pause for a minute and talk about one of our dominant forest species in the Pacific Northwest, Douglas-Fir, Pseudotsuga menziesi, giants of the Pacific Northwest, and one of the reasons why we have the most carbon-dense forests anywhere on the planet. We brought this up before, let's reiterate it. We've got three big acronyms that are really important for thinking about carbon cycling, and let's remember them. GPP, we're talking about gross primary productivity or photosynthesis. NPP, we're talking about net primary productivity, or the amount of carbon that's fixed into usable, actual plant biomass, leaves, stems, sticks, roots. And NEP, and this is the net change in carbon contained within an ecosystem over time. So this is accounting for decomposition processes as well as fixation processes by active plants. A young system, before we used Mount St. Helens, now we can use an open field, say, doesn't contain much carbon. A middle-aged Douglas-Fir forest starts to look like it contains more carbon, but it's structurally pretty uniform. And then we reach highest complexity and highest carbon storage in an older forest. This is true for other systems too. We could also, many of the concepts we're talking about now could be applied, for example, to Eucalyptus regnans forests in Tasmania and southeastern Australia. This is a photo of some of those older forests in Tasmania. Although their understories look very different superficially, these can be similar forests to some of ours. Here's an example table of plant carbon, soil carbon, and net primary productivity in the world's major biomes. And this kind of allows you to compare the areal extent of different biomes, the net primary productivity in those biomes, and then the plant and soil storage. And what you should notice right away is that tropical forests can really dominate in terms of NPP. This won't be true in terms of some other, like NEP, estimates of net carbon accumulation in systems, but it's certainly true in terms of the total amount of carbon that is fixed into usable biomass. But remember, we haven't accounted for decomposition yet. So plant carbon can be very high in terms of the total amount that's there, and soil carbon can be very high. Sometimes in the Pacific Northwest, we're talking about carbon density. And so it's really important to realize that carbon density, we're talking about carbon per unit area. And we're talking about it regardless of live versus dead carbon. And in the Pacific Northwest, we have some of the most carbon dense forests, but that doesn't mean that overall we contain the largest amounts of carbon on the planet. Undoubtedly, that's in tropical forests, no doubt about it. On a per area basis, we have extremely dense forests, especially because temperate rainforests of the Pacific Northwest have a relatively small footprint compared to many other types of forest systems. You'll see grasslands contain far less. Wetlands, which are often talked about in terms of how much soil carbon they have, are really important for carbon storage, but you see they're barely going to register for us in terms of NPP and plant carbon, depending on the type of wetland we're talking about. So they're going to have much less standing carbon, even if they have high soil storage. A few things to talk about, and then we'll move on from this. Photosynthetic capacity is often related to leaf area and soil resources. So leaf area in particular is a very big predictor of the amount of photosynthetic capacity that plants have. However, it's not just how much leaf area you have, it's also how those leaves behave and the amount of photosynthetic capacity, which fortunately, as we talked about earlier, is really governed by nitrogen. The larger amount of nitrogen you have, the larger amount of rubisco you have, and the larger CO2 you can fix. So high leaf nitrogen and high leaf area can combine to mean large photosynthetic capacity. Here's an example. In this graph, we've got measured leaf nitrogen from multiple systems and the net photosynthesis rate. Leaf nitrogen is a big determinant of photosynthetic capacity. If I wanted to predict the total amount of photosynthesis that's taking place in any system, it turns out that I can think about everything that affects photosynthesis, but some things affect photosynthesis more than others. So this image gives us relatively different sized arrows for different effects on photosynthesis. We've got leaf area, nitrogen, and season length really dominating controls on GPP in most ecosystems. And temperature, light, and CO2 are all important for determining photosynthesis, no doubt about it, but they just don't compare to season length. If you have a growing season that lasts 365 days a year, that is a tremendous impact on your total photosynthesis over that year. It's compared to a system where maybe you have 200 to 300 days of a growing season. Leaf nitrogen is the next important thing, and leaf area. What's cool about those three things is if those three things govern most of photosynthesis, most of gross primary productivity, the total amount of carbon sucked into an ecosystem, we can measure season length remotely, right? We know roughly what the season length is for any given system. We can measure it with weather stations, but it's in the weather data. Nitrogen and leaf area might be something that we can measure remotely. And if we can do that, then we can actually measure photosynthesis from satellites. Because leaf area is a good predictor of photosynthesis, looking at canopies from space provides us a lot of information. It turns out when sunlight strikes objects, of course, chlorophyll is going to absorb visible radiation and little near-infrared radiation. This is related to the amount of nitrogen, the amount of rubisco that's active in those leaves. So when we measure leaf area, combined with the quality of light that's reflected from those leaves, we can actually combine that to estimate the amount of photosynthetic potential or activity that's going on in those leaves. And we can differentiate that from bare soil surfaces. So now there's a whole host of satellite technology that's used to be able to do this. So you can Google this, you can look up MODIS or other instruments that are active on satellites right now. This technology has just taken off, so that now we have satellites that are attempting to do things like circle the entire, get imagery for the entire globe within a 24-hour period, so that we can collect reflectance data and imagery data from the entire globe every 24 hours or in short time periods, and then combine that together to get estimates of our amounts of gross primary productivity that are taking place anywhere in the globe in near real time. This is an older example now. This is 20 years old. But even 20 years ago, we were able to stitch together imagery from different time periods. In this case, this went from March 26th to April 10th in the year 2000. And within that time period, we had enough cloud-free, clear days and enough good imagery on the limited satellites that were up at that time to stitch together an image that gave an estimate of GPP based just on the reflectance data taken from space. And it's fairly accurate. The brown areas here represent places where spring has not fully sprung between March 26th and April 10th. And then, of course, the green to blue areas are places where spring is in full, running full tilt. Okay. Of course, there's a fair amount that we have to do to measure NPP that's different. NPP tends to be a half of GPP, but there's a fair amount that we have to do on the ground. I'm going to save that for a later time and close this out with saying these cycles are all clearly connected. And I hope this lecture helped kind of tie that together and show how interconnected these systems really are in terms of their element biogeochemistry. And hopefully it's just gotten you excited to go a little bit deeper into each of those elements, thinking about what governs them and what those cycles are like.